Hello everyone and welcome. My name is Priya Patel and I'm thrilled to share this presentation with you on the cell cycle, cell signaling, and their relationship to disease, including cancer. This presentation is designed to help you understand the vital processes that keep our cells functioning, how they sometimes go wrong, and what this means for diseases like cancer. By the end, I hope you'll have a solid grasp of all the processes and their importance in both biology and medicine. So let's get started. So let's begin by understanding why the cell cycle and cell signaling are so important. The cell cycle is like a careful planned schedule that cells follow to grow, replicate their DNA, and divide into two daughter cells. It ensures that the billions of cells in our body are constantly replaced. On the other hand, cell signaling acts as a communication network between cells. It allows cells to receive and respond to signals from the environment, whether it's nutrients, hormones, or even stress. When these processes are functioning well, they ensure proper growth and repair. But here's the catch: when they fail, the consequences can be severe. Dysregulation in the systems due to mutations or external factors can lead to uncontrolled growth, which is hallmark of cancer. Throughout this presentation, we will see how these two systems are interconnected and how they influence the development of cancer. Consists of four key phases, which we can see, that is G1, S, G2, and M. In the G1 phase, cells grow and prepare for DNA replication. Think of it as a cell gathering supplies for the next step. During the S phase, the DNA is replicated. This is like making an exact photocopy of all the genetic material. In the G2 phase, the cell prepares for the division by growing further and checking for any errors in the DNA. Finally, in the M phase, the cell divides into two identical daughter cells through a process called mitosis. But here's the fascinating part: cells don't just move blindly from one phase to another. There are checkpoints, like quality control stations, that ensure everything is in order before proceeding. Like, for example, if DNA is damaged, the cell will pause to repair it. If these checkpoints fail, however, cells may divide uncontrollably, which can lead to diseases like cancer. Moving forward, the smooth progression of the cell cycle depends on molecular regulators, specifically cyclins and cyclin dependent kinases or CDKs. Cyclins are proteins that acts as the cell's timekeepers. They bind to um, cyclin dependent kinases, activating them to push the cell into the next phase of the cycle. Then we have tumor suppressors like the retinoblastoma protein and P53. This protein acts as the cell security guards. For example, RB prevents a cell from prematurely entering the S phase by blocking certain factors until the cell is ready. P53, often called the guardian of the genome, pauses a cycle if it detects DNA damage. If the damage is irreparable, it triggers apoptosis or programmed cell death to prevent harmful mutations from the spreading. Now, when these mechanisms fail, either because of genetic mutations or environmental factors, cells may continue dividing despite errors, laying the groundwork for cancer. Now, let's talk about what happens when cells are damaged beyond repair. The body has mechanisms to ensure such cells don't persist. This is where apoptosis or programmed cell death comes in. Here how it works. Proteins like BAX and BAC promote apoptosis by releasing molecules that trigger cell death. On the other hand, anti-apoptotic proteins like BCL2 prevent cell death. While this is useful in healthy cells, cancer cells often exploit BCL2 to invade death. even when they are severely damaged. In some cases, cells would also undergo necrosis instead of apoptosis. Now, necrosis is an uncontrolled form of cell death, which can lead to inflammation. Cancer cells find ways to avoid both apoptosis and necrosis, allowing them to grow unchecked. Cell signaling is a complex network that coordinates how cells respond to their environment. Key players in signaling include MAPK pathways, which regulate cell growth, second messengers, which amplify signal within cell, cytokines and hormones, which you can see in the poster, which facilitate communication between cells, growth factors, which promote cell division. Now, in cancer, this pathway often becomes overactive. For instance, aggressive signaling in the MPK pathway can drive uncontrolled cell division. Now, understanding these pathways has led to the development of targeted therapies, which we will explore shortly. Moving forward, in cancer, the very signaling pathway that usually keeps cells in check become hijacked. That is cancer and dysregulated signaling. For example, mutation in the RAS gene activate the MAPK pathway, continuously driving uncontrolled growth. HER2 overexpression, often seen in breast cancer, amplifies growth signals. Targeted therapies like EGFR inhibitors and HER2 targeting antibodies are designed to block these pathways. However, cancer cells are clever and can develop resistance, which remain a major challenge in the treatment. Forward. So, how exactly does a cell cycle contribute to cancer? So, there are some of the key factors that influence this. First one is loss of tumor suppressors like P53 that removes brake on the cell division. Then the other one is overexpression of cyclines, like cyclin D speeds up the cell cycle. Overactive CDKs drive the cycle forward even when errors are present. To combat this, we have drugs like CDK inhibitors. Palbociclib, for instance, is used in breast cancer treatment to slow the cell cycle and prevent tumor growth. The cell cycle and cell signaling are deeply interconnected with the integration of cell cycle and signaling cancer. For example, DNA damage activates ATM and ATR protein, which pause the cell cycle through CHK1 and CHK2 signaling, which is mentioned that is leads to CHK1 and CHK2 activation. Moving forward, we can see in the path that meanwhile pathways like PI3K and AKT promote survival by inhibiting apoptosis. Then there is MAPK signaling on the other hand that drives the production of cyclin D, pushing cells into the S phase. Now, cancer disrupts this balance, leading to unchecked division and survival. Understanding this pathway has revolutionized cancer treatment. Here are a few approaches that are therapeutic implications. First one is CDK inhibitors, which helps in the halt of cell cycle progression. Then the other one is apoptosis inducing drugs, which directly targets proteins like BCL2. Then there are personalized medicine, where treatments are tailored based on an individual's genetic makeup. There are combination therapies too, which use multiple drugs to overcome resistance. Now, this each approach targets specific weakness in cancer cells, making treatments more effective. Current research, the future of cancer treatment look promising. Researchers are exploring artificial intelligence to predict drug responses and optimize treatments. They are also using emerging biomarkers to identify cancer earlier. The most prominent is CRISPR technology for precise gene editing. There are combination therapies to improve outcomes by targeting multiple pathways. Now, these advances are bringing us closer to a future where cancer is no longer a devastating diagnosis. Thank you for your attention. I hope this presentation has given you a deeper understanding of the cell cycle, cell signaling, and their role in the cancer. If you have any questions, I would be happy to answer them in the mentioned email in my YouTube video. Thank you so much.